Good morning everyone. Today in this session I am going to discuss regarding the structure of proteins. The structure of proteins we can discuss at 4 different levels. First one is the primary structure, second is the secondary structure, third is a tertiary structure, fourth is the quaternary structure. First one is the primary structure of proteins. Here the primary structure of proteins is given based upon the number of amino acids involved in the formation of a polypeptide chains. Here the number of amino acids involved gives a primary structure, as well as here the amino acids which are involved in the polypeptide chain formation, the sequence is also very important. For example, if I take this primary structure, it is made up of first as glycine, second is alanine, third is leucine. This is the sequence of the polypeptide chain. Amino acids are in the sequence of glycine, alanine and leucine. Here these three amino acids, next it will repeat as glycine, alanine as well as leucine. Here they are arranged in a regular sequence. Each protein is made up of one or more polypeptide chains in which the amino acids are arranged in a specific sequence. Here the sequence is also important, as well as the number of amino acids involved is also very important. If one amino acid sequence is changed, here after alanine instead of lysine, if another amino acids, for example isoleucine is being placed, then this protein becomes a different type of proteins. Each protein is having a specific number of amino acids. They are linked together through a peptide linkage. Here the sequence, if it changes, even one amino acid also sequence changes, it becomes a different protein. What does this primary structure gives here? Primary structure, it gives the sequence of amino acids as well as the number of amino acids present in a polypeptide chain. Here in the proteins, the polypeptide chain may be one or more than one polypeptide chain. This gives the primary structure. Next comes the secondary structure. What is the role of this secondary structure here? The secondary structure gives the shape of the polypeptide chain. The shape of the polypeptide chain is due to the formation of hydrogen bonding. This secondary structure is formed by the folding of a polypeptide chain, folding of a backbone of a polypeptide chain. Here there is a regular folding of polypeptide chain takes place in two different types. One is the alpha helix structure and another is the beta pleated structure. The regular folding of the amino acids in a polypeptide chain leads to the formation of a secondary structure. In secondary structure, two types of folding is possible. One is alpha helix structure and another is a beta pleated structure. Here if you see, there is an R groups are present. If R groups are having very small groups, then the polypeptide chain is coiled in such a way that it will form a beta pleated structure. If R group is very large size, then the polypeptide chain, it will coil by itself to form a alpha helix. Alpha or beta, it depends upon the R group. If R group is large, the polypeptide chain will coil at the backbone to form alpha helix structure. If R group is small, it will form a beta pleated structure. Here the secondary structure of proteins explains regarding the shape of a polypeptide chain. The shape depends upon the hydrogen bonding. The secondary structure of proteins, in that first comes here the alpha helix structure. Here these polypeptide chains may be one or more than one polypeptide chain may be present. The backbone of the polypeptide chain is coiled in such a way that it is coiled in a right-handed screw. If the polypeptide chain coils in a right-handed screw, it will form a alpha helical structure. If the polypeptide chains coils in a right-handed screw, it will form a alpha helical structure. Alpha helix structure is formed by the coiling of a polypeptide chain in a right-handed screw. It will form as an alpha helical structure. Here, if you observe the coiling, the twisting takes place. Here, the twisting takes place. Where the twisting takes place? The next atom is the carbonyl group. Adjacent carbon to the twisted alpha helix is the carbonyl group, which will form a hydrogen bonding with the NH group of the another twisted form. CO-NH hydrogen bonds are formed. Due to this hydrogen bonding between the carbonyl group of one amino acids adjacent to the twisted position with the NH group of the another amino acid, it will form a hydrogen bond. Due to the formation of hydrogen bond between CO of one amino acids and the NH of the amino acid, the alpha helix is obtained. Here it is obtained by the right-handed screw. If the polypeptide chains, if R group is small, the polypeptide chains, it will form a beta pleated sheet or beta pleated structure of proteins. In beta pleated structure, here the polypeptide chains, they are stretched out. After stretching out the polypeptide chains, they are placed by side, and these stretched polypeptide chains, they are linked by a intermolecular hydrogen bonding. The carbonyl group of one polypeptide chain, it is linked with the hydrogen atom of NH of another polypeptide chain through a intermolecular hydrogen bonding. They are linked by intermolecular hydrogen bonding. These polypeptide chains, they are stretched out, they are placed side by side, and they are linked by intermolecular hydrogen bonding, and it will form a beta pleated shape. Beta pleated is nothing but the pleats are formed. Pleats are nothing but the foldings. Foldings are known as the pleat structure. Since they are placed side by side, it looks like a beta pleated sheet. Here R group is small in case of beta pleated sheet, whereas in case of this alpha helix, R group is large. Example for this alpha helix structure is the keratin. Keratin protein, it will form a alpha helical structure, and beta pleated shape, it is seen in case of silk protein fibron. Silk protein fibron. fibrion is the example for the beta pleated sheets. Secondary structure gives the arrangement of the polypeptide chains. It may be alpha helical structure or it may be beta pleated sheets. Third comes here the tertiary structure. Tertiary structure explains regarding the overall shape of a polypeptide chains. Here it may be one polypeptide chain or maybe two polypeptide chains. Tertiary structure, it is having two types. One is the fibrous structure. And another second one is the globular structure. The polypeptide chains are coiled among themselves to form a spherical shape. It will form a globular structure. If the polypeptide chains forms a thread-like structure, then it is known as a fibrous structure. Fibrous and globular is named based upon the shape of these chains. If it is a thread-like structure, then it is known as the fibrous proteins, fibrous. If it is a globular spherical means it is known as the globular structure. Tertiary structures gives the overall shape of a polypeptide, fibrous or it may be globular. Here the secondary as well as the tertiary structures of the proteins is maintained due to the following forces. One is due to the van der Waals interaction. In secondary as well as tertiary proteins, they are regulated by the van der Waals interactions, hydrogen bonding, and due to electrostatic force of attraction, as well as these shapes are due to the disulfide bonds. These four are responsible for the shape of a secondary as well as the tertiary structure of proteins. In the annual exam, you get for 1 mark which are the forces responsible for the shape of a secondary as well as tertiary structure means. Here first is the hydrogen bonding, second is disulfide bonds, third is the van der Waals interaction, fourth is the electrostatic force of attraction. These are responsible for the secondary and tertiary structure of a proteins. Then the last one comes here, the quaternary structure of a proteins. If you see the quaternary structure of a proteins, here it is made up of more than one polypeptide chains. Here each polypeptide chain is having special arrangement with that of the another polypeptide chain. Then the structures are known as quaternary structure. Each polypeptide chain is having a unique structure with that of the another polypeptide chains. It will form a quaternary structure of proteins. This is regarding the structure of proteins. After structure of proteins, next comes here denaturation of proteins. Denaturation of proteins. Proteins, when they are subjected to heat or they are treated with acids or on treating with bases or with salts. Proteins, when they are treated with acid, bases, salts or on heating, here the protein loses its biological activity. The process by which the proteins loses its physical and biological activity on applying heat, acids, bases or salts. This process is known as denaturation of proteins. Here, if I take egg, the white part of the egg, if I heat the white part of the egg, what happens? It becomes solidified. The physical changes takes place, as well as here its biological activity also changes. Then heating of white egg is an example for the denaturation of proteins. If I take milk and if I add the curd to that, what happens? The curdling of the milk takes place. You know that the bacteria lactobacillus, which is present in the milk, is responsible for the curdling of the milk. After curdling of the milk, what happens to the milk here? Its physical property changes, as well as here its biological activity also changes. So, curdling of milk is an example for denaturation of proteins. Another one is the heating of white egg is an example for denaturation of proteins. When you subject this protein to either heat, acid or base, it loses its biological as well as its physical property. Then it is known, the process is known as denaturation of proteins. Here, once you see the curdling of the milk takes place, can we reverse it back into milk here? Is it possible? No. So, denaturation of proteins, these are irreversible process. And what happens to the entropy here? Entropy always increases. Here, the increase in entropy is always seen in case of the denaturation of proteins. In the competitive exam, they ask that what happens to the term entropy during the denaturation of proteins. Entropy always increases, and here this denaturation of proteins, it is a irreversible process. Curdling of milk and the heating of white egg are the examples for the denaturation of proteins. Here, once the denaturation of proteins takes place, what happens to the structure of proteins? Whether there is a change in the structure of proteins? Here, the primary structure of proteins, it remains same. What does primary structure proteins gives? It gives the number of amino acids as well as a sequence of amino acids. It remains same. Only there is a change in the secondary and tertiary structure of proteins. In the exam, they ask for 1 mark question. What happens on denaturation of proteins? So, which structure remains intact? Intact means same. Primary structure remains the same. Only there is a change in the secondary as well as the tertiary structure of proteins. Next comes here the enzymes. Enzymes are nothing but these are the proteins. Enzymes are proteins which act as a catalyst in biochemical reactions. What are enzymes? Enzymes are proteins which act as a catalyst in biochemical reactions. Here, example for enzymes is maltase is an enzyme, lipase is an enzyme, amylase, sucrose, pepsin, trypsin. These are all the example for enzymes. Enzymes are very active at 37 degrees Celsius. Enzyme activity is maximum at 37 degrees Celsius. 1 mark question, when the enzymes are active here, maximum activity is observed at 37 degrees Celsius. These enzymes are very specific in nature. Here, for example, the reaction is taking place in presence of maltase, means instead of maltase, if I add the lipase, there is no any reaction takes place. The reaction takes place only in presence of maltase. If other enzymes is also present, it does not interfere in the chemical reactions. A specific enzyme is responsible for a specific process to take place. Hence, enzymes are very specific in nature and they are having maximum activity at 37 degrees Celsius. If the temperature increases, here its activity goes on decreasing, and when it reaches the temperature around 65 degrees Celsius, here these enzymes are inactive. They are inactive or ineffective at 65 degrees Celsius or above 65 degrees Celsius. The enzyme activity is not observed here. So, when it is high here, when it is 37 degrees Celsius, the activity of enzyme is very high. As the temperature goes on increases, the activity of enzyme decreases and it is totally ineffective when it reaches 65 or above 65 degrees Celsius. These enzymes are required in very small amount. Even small milligrams or micromilligrams of enzymes is enough to carry out the reactions. And why we use these enzymes? Because these enzymes, they increase the rate of a reactions. How the rate of a reaction is increased due to the presence of enzymes means, you know the activation energy EA. EA is known as the activation energy. This much amount of energy is required to convert the reactant into the product. EA is known as the activation energy. So, each reaction to convert reactant into product, it should have the specific activation energy. When it crosses over its specific activation energy, the reactants are converted into product. In presence of enzymes, the activation energy decreases. Enzymes decreases the activation energy. The conversion of reactant into product takes place in presence of small activation energy. Here, due to the decrease in activation energy, EA value decreases in presence of enzymes. So, maximum number of reactants are converted into products. Hence, rate of a reaction also increases. Here, how much rate of a reaction increases means, here the rate of a reaction increases in presence of enzymes is manifold times. Manifold times the reactants are converted into product. Manifold times means 10 to the power of 6. Rate of a reaction increases by 10 to the power of 6 times in presence of enzymes. Hence, enzymes are used as a catalyst for various reactions to increase the rate of a reaction. Why it increases the rate of a reaction? Because it decreases the activation energy. Maximum number of reactants are converted into products. Rate of a reaction is increased by manifold times, that is 10 power 6 times. Let us see some of the reactions which are taking place in presence of enzymes. For example, maltase. In presence of enzyme maltase, sucrose produced on here, sucrose on hydrolysis produces glucose and fructose. In presence of amylase as an enzyme. Here you can use amylase or salivary amylase as an enzyme or sucrase as an enzyme to convert sucrose into glucose and fructose. Maltose is converted into glucose units, 2 glucose units in presence of maltase as an enzyme. Only maltase will convert maltose into 2 glucose units. Lipase. Lipase, it converts oils and fats into glycerides and fatty acids. Lipase is an enzyme responsible for the conversion of oils and fats into glycerides and fatty acids. Pepsin and trypsin. These are used in the hydrolysis of proteins into alpha amino acids. These are some of the examples of the enzymes which are involved in the chemical reactions. Enzymes are very specific in nature. They are maximum, its activity is maximum at 37 degrees Celsius. As the temperature goes on increasing above 65 degrees Celsius, they are ineffective takes place. Enzymes is very specific. Here a specific enzyme only converts the specific reactant into product. If other enzymes is also present, but here it is not involved in the chemical reactions. Here enzyme, it is having two parts. It is having two components. One is the protein part and another is the non-protein part. The protein part, the component of the protein part is known as apoenzyme. What is an apoenzyme? The protein part of the enzyme is known as apoenzyme. For 1 mark question you get what is apoenzyme. The protein part of the enzyme is known as apoenzyme and the non-protein part of the enzyme is known as coenzyme. The non-protein is known as coenzyme or cofactor. In the competitive exam also they ask you what is apoenzyme. Apoenzyme is the protein component of the enzyme. Non-protein component of the enzyme is known as coenzyme or it is known as cofactor. In the next session we discuss regarding the vitamins, hormones. Thank you.